Alright, ji, Assalamu alaikum everybody. How are you all? I hope that you all are doing well. Welcome or welcome back to the class. So today basically we are going to be looking at the AS level biology 9700 past paper, which is from the year May June 2023, paper 2-2. So we are going to be discussing the year May June 2023, paper 2-2 of the AS level biology 9700. Let's start off by discussing question number 1 over here. Now question number 1 over here is from the transport in mammals, which is a very high yield chapter. Now let's start off with this question and let's see that what the examiner is asking us in this question. The sinoatrial node SA node and the atrioventricular node AVN have an important role in the control of the cardiac cycle. The timing of the atrial and the ventricular systole and diastole must be controlled so that the blood passes through the heart efficiently. Now part A is saying that figure 1.1 is a summary of the blood flow through the right side of the heart during one cardiac cycle. 3 boxes in figure 1.1 are not completed. Complete the boxes 3, 5 and 7 in figure 1.1 using only the term systole and the diastole. So basically they have given us the stages of the cardiac cycle 1, 2, 3, 4, 5, 6, 7 stages of the cardiac cycle. The examiner has given us the 7 stages of the cardiac cycle and they have specifically talked about the right side of the heart over here in these 7 stages, and we just have to fill these blanks by writing the systole or we have to write the diastole. Now, so if I give you an overview of it, so we all know that during the cardiac cycle what happens is that the atria and the ventricles, they both contract and relax in a very much coordinated way, right? So that the cardiac cycle can, or you can say the blood can flow efficiently through the heart. Now let's begin reading the stages over here. Blood arrives at the heart in the superior vena cava and the inferior vena cava. Then what happens is that the blood passes into the right atrium. And how is the blood going to pass into the right atrium? If you guys are going to tell me, then obviously as the blood is going to pass into the right atrium, let me copy down this diagram over here, which is in my lecture notes of the transport in mammals, so you guys can have a better idea about it. Alright. So now what is happening, that the blood is passing from the superior vena cava and it has entered into the right atrium of the heart, right? Alright, now then they are saying that atria and ventricle, and then step 4 is that blood is forced into the right ventricles. So how is basically the blood going to be forced into the right ventricles? Obviously it is going to be forced into the right ventricle when the right atria is going to contract. And how can we say that the right atria is going to contract? If the right atria is going to contract, we are going to say that the atrial systole is happening. And obviously we all know that during the atrial systole there is the ventricular diastole. The ventricles will stay relaxed because they are receiving the blood. So the ventricular diastole will be there. Then when the blood has reached the right ventricles, then what is happening, that the blood is leaving the right ventricles to enter into the pulmonary arteries, right? It is going to leave the right ventricle and is going to enter into the pulmonary arteries. So how is the blood going to leave the right ventricle and enter into the pulmonary artery? We all know that it will happen only when the ventricles are going to contract, right? So the ventricles are going to contract, so we are going to write ventricular systole is going to happen, and when the ventricles are going to contract, it is going to pump the blood into the pulmonary artery on the right side and the aorta on the left side. And obviously the atria will stay in a relaxed state, it is going to stay in the diastole. Then what is happening, that then what will happen, that when the blood is going to leave the right ventricle and enter into the pulmonary artery, then finally both the chambers of the heart are going to relax to allow the heart to refill with the blood. So we are going to write that atrial and ventricular diastole will happen. The whole heart is going to relax so that the heart is refilled with the blood. So basically this is how you are supposed to get yourself done with question number 1 part A. I hope that this is clear to you guys. Now let's move on to part B over here. Now moving on to part B over here. Part B over here says that impulse sent out by the sinoatrial node passes to the AV node where there is a short delay. With reference to figure 1.1, explain why is it important for the control of the cardiac cycle that there is a short delay at the AV node after the impulse have been sent out by the SA node over here. Alright, so if I copy down this image once again for our reference over here. So we know that the conducting system of the heart is basically also known as the nodal system, right? The impulse is generated by the sinoatrial node and the impulse travels the impulse of electrical excitation travels throughout the atria and both the atria contracts and then the impulse goes to the AV node, right? And over here in the AV node what happens is that the electrical impulse delays for 0.09 or 0.1 seconds. So they are saying that why the AV node has been delayed, why the electrical impulse have been delayed at the AV node. So what is the possible answer for this, that why there is a delay of the electrical impulses at the AV node? It is because to allow the atria to fully contract, so that the atria can fully contract and it can fully empty its blood completely into the ventricles, right, before the ventricle begins to contract. Also I have discussed this in my lecture notes as well, that why the atria why is there a delay in the contraction. Also I have discussed it in my lecture notes as well, that this delay, you guys can see over here, I have also written the reason, that this delay allows the atria to fully contract before the ventricles and hence to prevent the simultaneous contraction of the atria and the ventricle. So this is basically the answer to part B over here. I hope that this is clear to you guys. Alright, so now moving on to part C over here. Part C over here says that changes in the blood pressure occurs in the heart during the cardiac cycle. These changes causes the opening and closing of the bicuspid and the tricuspid valves, which are the atrioventricular valves. Alright, so if I talk about the bicuspid valve, so bicuspid valve are the valves which are found in the left side of the heart and which are also known as the mitral valves. Also the tricuspid valves are the valves which are found at the right side of the heart. Alright, the question further continues, and the aortic and the pulmonary semilunar valves. Explain how the blood pressure changes causes the opening of the tricuspid valve, causes the opening of the pulmonary valve and helps to flow the blood through the heart. Alright, so they are also talking in reference to the right side of the heart over here in this part as well. So we all know that the blood moves through the heart because of the pressure differences. Now when the atria is going to contract, let me again basically move this diagram over here. Now what is going to actually happen, that when the atria is going to contract, the pressure inside the atria is going to rise above that of the ventricles over here, obviously because it is contracting. When the atria is contracting, so the pressure inside the atria will rise above that of ventricles and the tricuspid valve will open and the blood will flow into the ventricles. And then what will happen, that the ventricles are going to contract. And when the ventricles are going to contract, what is going to happen, that the pressure of the ventricles is going to exceed the pressure of the atria, as well as it is going to exceed the pressure of the pulmonary artery, and hence what is going to happen, that the pulmonary semilunar valves are going to be opened and the blood will be ejected from the pulmonary artery into the lungs, right? So the blood from the right ventricles will be ejected into the pulmonary arteries, and the pulmonary arteries will basically flow them towards the lungs. and then what is going to happen, that as the ventricles are going to relax, the pressure is going to fall again, closing the semilunar valve, and it is going to prevent the backflow of the blood, obviously. So these pressure changes overall is basically keeping the one way flow of the blood smoothly. So hence the blood flows from a region of high pressure to a region of low pressure, and the valves between them, the tricuspid and the pulmonary valve, ensure the one way flow of the blood, and it prevents the backflow of the blood, maintaining the efficient circulation of the heart, right? So how are we basically supposed to write its answer? We can write the answer in the way that the tricuspid valve open when the pressure in the right atrium exceeds the pressure in the right ventricle. Obviously, because the right atrium is contracting, so the pressure is going to be high in the right atrium. And then when the blood is going to enter into the right ventricle, what is going to happen, that when the pressure in the right ventricle becomes higher than the pressure in the pulmonary arteries, so the pulmonary valves are going to get opened, and the blood is going to flow from the right ventricles to the pulmonary artery. So hence the blood flows from the higher pressure to the low pressure inside the heart, and the valves in the heart also ensures the one way flow of the blood. It prevents the backflow of the blood. Right, so this was all about question number 1 over here. I hope that it is clear to you guys. Now let's move on to question number 2 of this paper. Question number 2 over here says that figure 2.1 is a photomicrograph of a longitudinal section through a root tip. Two different regions are visible. One, they are saying the root apical meristem, and the root cap is visible over here. Then further in the question they are saying that cells in the root cap synthesize a gel-like sticky secretion known as the mucilage, which is important in reducing the friction between the soil and the growing root. It is composed mainly of the polysaccharide and also contains some amino acids and the enzymes. So this is basically figure 2.1 over here, which is showing us the photomicrograph of the longitudinal section of the root tip over here. And over here you guys can see, for example, this area over here is representing the root cap. This area, if you guys can see over here, is representing the root cap, and the area above over here is basically representing the root apical meristem cells. It is the root apical area. This, the one I am highlighting with the pink one, is the root apical area with the root apical meristem cells. Alright, so now let's move on to part A over here. What is the part A of this question basically saying to us? It is saying that describe three differences visible in figure 2.1 between the root apical meristem cells and the root cap cells. So basically we have to compare the cells in the root cap, and we have to compare the cells in the apical region of the root. Now if we start to compare the cells in the root cap and the cells in the root apical region, so if you see over here, you guys can see different phases of the mitosis shown over here. The chromosomes are also visible to us. It means that the root apical meristem cells show some cells in the mitosis. Whereas if I talk about the root cap cells over here, so root cap cells over here is just having a visible nucleus over here, and it is not showing any mitosis because the chromosomes are not that much visible over here as they are over here in a thread-like form, right? So the first difference you can write over here is that the root apical meristem cells, this is the short form which I will be using for the root apical meristem cells, but make sure that when you are solving this in your papers, in your actual examinations, then do write the full form. So the first difference we discussed that the root apical meristem cells show some cells in the mitosis, whereas the root cap cells show no mitosis, right? And then let's talk about the other difference over here. Now if I talk about the other difference over here, so you guys can appreciate this as a cell, right? You guys can appreciate this as a cell. So you guys can see over here that the root apical meristem cells are smaller with the larger nuclei, right? Relative to the cell size, the nuclei is larger, whereas the cell is smaller but the nuclei is larger as compared to the cell size. That is why we are saying that the cell is smaller, because the size of the nuclei is actually larger relative to the cell size. Whereas if I go to the root cap cells over here, root cap cells, you guys can see that in the root cap cells what is happening, that while the root cap cells are larger, for example you can take this cell, while the root cap cells is larger, but it is having a smaller nuclei relative to the cytoplasm. Look over here also, you guys can see over here also in this cell, the cell size is larger but with a smaller nuclei relative to the cytoplasm, you guys can say. Okay, so this is the next difference which we talked about. The root apical meristem cells are smaller with larger nuclei relative to the cell size, while the root cap cells are larger with the smaller nuclei relative to the cytoplasm over here, right? So this is basically the next difference, the second difference we talked about. Now if I talk about the third difference over here, now the third difference you guys can see, if I make it more magnified to you guys, then you guys can see over here that the cells in the root apical region are very much regularly arranged, like in a row, and they are tightly packed together as well, and they are regularly arranged and tightly packed together in a row as well. Now if you see the root cap cells over here, so you guys can see that it is very much irregular in shape. One is like this, the other one is like this, the other one is like this, and also they are loosely arranged as well. So you guys can say that the next difference over here, you guys can say that the root apical meristem cells are regular and tightly packed in the rows, while the root cap cells are irregular in shape and they are loosely arranged, right? So this is basically how you are going to tell the visible differences between the root apical meristem cells and the root cap cells. I hope that this is clear to you guys. Now let's move on to part B over here. Alright, so moving on to part B over here. Part B over here says that mucilage acts as a glue to bind tiny soil particles together. If you remember, what was the mucilage? Mucilage was a sticky secretion that was being released by the root cap cells, that was being synthesized by the root cap cells and was being released in the soil. Now they are saying that mucilage acts as a glue to bind tiny soil particles together, forming the small clumps close to the root. These small clumps help to maintain the soil water around the root tips and prevent the loss of the water. Then further they are saying that with reference to the cohesive and the adhesive properties of the water, suggest and explain how the formation of the small clumps of soil help to maintain the soil water around the root tip. So now let me make you guys visualize this all. Like for example, these are the root tips, right? And the mucin that has been released is basically made these sort of the soil clumps. Now they are saying that how does the cohesive and the adhesive properties of the water is basically helping to maintain the soil water around the root tips. So first of all, I have to tell you guys that what is the cohesive and the adhesive property of the water. So what does it mean by the cohesive property of the water? So the cohesive property of the water is that the water molecules stick to each other, right? When the two water molecules stick to each other, it is by the cohesive forces, it is the cohesion. And when the water molecules stick to the other molecules or the other surface, so that is the adhesive forces. For example, the water molecule sticks with the soil. This is the soil particles. The water molecules stick with the soil particles. So this is the adhesive property, right? So this is what the cohesive and the adhesive property of the water is. Now coming back to the question, they are saying that we have to explain that how these properties are actually going to maintain the soil water around the root tip. So when the mucilage is going to bind the soil into the small clumps, so what is going to happen, that the adhesion is going to make the water cling to the soil, means that it is going to attach the water to the soil clumps and the mucilage surface as well, because obviously the mucilage surface is the one that is forming the small clumps of the soil. So this adhesion property is basically going to allow the water to be held in the small clumps of the soil near the root tips. And then what is going to happen, that this is adhesion. Then what the cohesion is going to do, that the cohesion is basically going to keep the water molecules connected together, right? So now because of these combined forces of the adhesion and cohesion, by the adhesion the water molecules will adhere to the soil particles of the clumps and the mucilage, and by the cohesion what is going to happen, that the water molecules will stay connected to each other, and by this these combined forces are going to help to trap the water in the spaces between the soil particles, right? These combined forces are going to help to trap the water in spaces between the soil particle. So how is basically these adhesive and the cohesive forces allowing as well as helping to maintain the soil water around the root tip? We have talked that this adhesive forces and the cohesive forces basically allows the water to be held in the small clumps of the soil near the root tip. So these cohesive and the adhesive forces are going to retain the water around the root, and it is going to prevent the water from draining away and maintaining a constant, or you can say a stable water supply for the absorption. So this is basically how you are supposed to answer this question. Now moving on to part C over here. Part C over here says that enzymes present in the mucilage catalyzes the breakdown of organic compounds in the soil. This increases the presence of the mineral ions in the soil. State the term used to describe enzymes that act outside the cells that synthesize them. So the enzymes that act outside the cell that synthesize them, outside the cell. If it is outside the cell, it is extracellular enzymes. And if it is inside the cell, if it is acting inside the cell, it is intracellular enzymes. So if the examiner has asked one question of outside the cell, in the upcoming years he can also ask that the enzymes that connect inside the cells as well. So you should know about that as well. However, coming back to the answer of this question, the answer of this question will be extracellular enzymes, right? Extracellular enzymes. Now moving on to the part D over here. Part D over here is relatively a very simple part. The polysaccharides and amino acids present in the mucilage are a source of nutrients for the soil microorganisms that live in the area surrounding the root. Some of these microorganisms can break down soil compounds to release the mineral ions. Then further they are saying in part 1 that explain what is meant by a polysaccharide. So what is meant by polysaccharides? So if I talk about the polysaccharides, so we all know that polysaccharides are the large carbohydrates which are made up of the monosaccharides, right? Wait, let me write that down. So we all know they are the large carbohydrates, right, which are made up of the monosaccharides. These are the key terms which I am writing over here, which are made up of the many monosaccharide monomers. You guys can say, for example, glucose, and they are linked together by the glycosidic bond. Many monomers, right? What is a polysaccharide? Is made up of it is a very large carbohydrate molecule which is made up of the many monosaccharide monomers which are joined together by the glycosidic bond in a reaction which is known as the condensation reaction. So this is basically how you are supposed to write the answer of the polysaccharide. Let's write down its ideal answer as well, that it is a polymer of many monosaccharides, which is the simple sugar units. You all know that monosaccharides are the simple sugar units. and they are joined together by the glycosidic bond. Also, it consists of the repeating sugar units forming the large macromolecules such as starch and the cellulose. About the polysaccharide, if I tell you a little more detail about the polysaccharide. So polysaccharides, there are two types of the polysaccharide. There is a structural polysaccharide which consists of the cellulose, and then there is a storage polysaccharide which is glycogen and starch. Starch is found in the plants. Also, the cellulose is also found in the plant cell wall. Glycogen, however, is a storage polysaccharide in the humans. Right, so basically this is how you are supposed to answer this question, and this is some detail about this part of the question that we discussed over here. Alright, so now moving on to part 2 over here. Part 2 over here says that the soil microorganisms use its amino acid to synthesize the protein. All the 20 different amino acids that are present in the protein have the same general structure. Alright, so we know that almost all of the amino acids are having a general structure. Also, if I go back to my class notes of the biological molecules to show you guys the general structure of the amino acids that we discussed in our lecture. So this is the general structure of the amino acid. We are having a central carbon which is attached to the amine group, and then the carboxylic acid group or the carboxyl group and the hydrogen atom, and then there is this R group over here. Now all of the variability actually depends upon the R group over here. Let me paste it over here. Now this is the general structure of the amino acid: the carboxyl group, the amine group, the hydrogen atom and the R group. What is the R group basically? R group is the variable group, and it can be any group, right? It is a variable side chain. Over here it can come CH2OH or it can come CH3. So this is basically the R group, which is a variable group, or you can say it is a side chain as well. Now they are saying that draw the general structure of an amino acid in the space provided. So this is the general structure of the amino acid already drawn, and use this drawing to explain how is it possible to have many different amino acids. So how are we basically going to use this image, this diagram, to show that we are having many different amino acid because of this R group over here. Because of this R group present over here, we guys can show that there are many possible different amino acids. How? Because we know that the R group, or we can say the R group or the side chain, is variable, which means that the different amino acids can have the different R groups. Different amino acids can have different R groups, giving each unique properties, giving each amino acid the unique properties, right? So this is basically about this part 2 over here. I hope that this is clear to you guys. So this question is actually a very lengthy question. Now let's finally move on to the last part of this question over here, the part 3 over here. Now the part 3 over here is saying that mineral ions are usually present in the soil in very low concentrations. The action of the mucilage enzyme and soil microorganisms can help to increase the presence of the mineral ions. Okay. Root hair cells are specialized for the uptake of these mineral ions and for the absorption of water from the soil. Suggest and explain how the presence of the mineral ions in the root hairs can increase the absorption of water by the root hair cells. So this is basically the diagram which is showing us the root hair cell. For example, let's draw the soil around it. This is the soil around it, okay? Now this is the soil around it, which is having some mineral ions over here, right? Which is having some mineral ions over here. Now they are saying that how the presence of the mineral ions in the root hair cells can increase the absorption of the water by the root hair cells. So how you can say this, that the mineral ions, for example you can say like nitrate or the potassium, when they dissolve in the cytoplasm of the root hair cells, so they increase the solute concentration in the cytoplasm, right? The solute concentration in the cytoplasm increases when the mineral ions are there. Mineral ions increase the solute concentration of the cytoplasm of root hair cells, and by this what happens that the as the solute concentration of the cytoplasm of the root hair cells increases, so similarly and simultaneously the water potential of the cytoplasm of the root hair cell decreases, right? So what will happen, we know that the osmosis occurs from a region of high water potential to a region of lower water potential. Let's mention this as well, that osmosis occurs from a region of high water potential to a region of low water potential. So obviously the water is going to enter the cell by the osmosis, right? The water is going to enter the cell by the osmosis, and this process will actually maintain a continuous uptake of the water. So this is basically how you are going to explain that how the presence of the mineral ions in the root hair cells can increase the absorption of water by the root hair cells, right? I hope that you got the answer, that when the mineral ions are going to enter the root hair cells, they are going to lower the water potential of the cytoplasm inside the root hair cells, and this is going to create a steeper water potential gradient between the soil and the root hair cell. So as a result the water is going to enter the cell by the osmosis, right? So this is basically how you are supposed to answer this part of the question. I hope that this question is clear to you guys. I have tried my level best to explain it to you guys. I hope that it makes sense and it is clear to all of you. Let's now move on to question number 3 over here, which is from the chapter of the immunity, which is the last chapter of your AS level biology. The question says that cells of the immune system respond to the presence of the non-self antigens. Now there are two types of antigens in the body. One is the self antigen and the other one is the non-self antigen. Now non-self antigen is basically identified as the foreign antigen, and the self antigen of the body is basically identified as the body's own cells antigen, right? So the self antigen is not that harmful, but the non-self antigen, if the body recognizes an antigen as a non-self antigen or a foreign antigen, then it is very much dangerous and it will basically initiate an immune response. Now part A over here says that outline the features of the non-self antigen. Now if I tell you more about the antigens over here, or more specifically about the non-self antigens, so antigens are usually the proteins, the glycoproteins that are found on the surface of the pathogen, infected cells or any foreign cells, right? Means that they are the foreign molecules which are often found on the surface of the pathogens, infected cells or the foreign cells. And also you guys can say that they are specific in shape, because antigens are very much specific in shape because they have to complement with the receptors of the beta lymphocytes or the T lymphocytes, right? So they can have the specific shape that is complementary to the antigen receptors on the lymphocytes or the other binding sites of the antibodies. Also, because this is a non-self antigen, it is the antigen that is recognized as a foreign antigen, so it is going to trigger the immune response as well. And when the immune system or the immune response will get triggered, so more antibody production will be there, there will be more lymphocytes activation, there will be more phagocytosis. So this is basically what the triggering of the immune response actually means. So I hope that this is clear to you guys. Moving on to part B over here. Part B over here says that 4 different types of the cells of the immune system are shown in the table 3.1. Complete table 3.1. Use a tick if the description applies to the named cell of the immune system. Use a cross if the description does not apply. Now they have given us a few of the cells of the immune system. That includes the beta lymphocyte, that includes the plasma cell, that includes the T helper cells and the T killer cells. Now let's read the description of these cells and let's see that the description of the cell matches which of these cells, right? So now let's start off with the first description, able to go through a number of cell cycles, which means the clonal expansion. So if I talk about the beta lymphocytes over here or the T lymphocytes, so they can divide rapidly, which is the clonal expansion, to form the plasma cells and the memory cells in case we are talking about the beta lymphocytes. And if we are talking about the T helper cells and the T killer cells, so they can also go through the repeated cell division for the clonal expansion. So beta lymphocyte, T helper lymphocytes, T killer cells and the plasma cells cannot go through the clonal expansion, right? Then further main role is to secrete the cytokines during the immune response. So basically the cytokines, what are the cytokines? Cytokines are basically the chemicals that are released by the T helper cells over here. They are the chemicals that are released by the T helper cells. And what does the cytokines actually do? They activate the other cells of the immune system, right? They activate, for example, the B cells, the T killer cells. They are going to activate the other cells of the immune system by releasing the chemical which is known as the cytokine. Now further, the third description says that present during the primary immune response to the virus. Now the virus word over here is very much important. So the cell that basically destroys the virally infected cells of the body, that is the T killer cell over here, which is also known as the cytotoxic T cell. It basically kills the or destroys the viral infected cells of the body or the virus infected cells of the body. So present during the primary immune response to a virus, obviously T killer cell, the cytotoxic T cell, because it is going to destroy the virus infected cell of the body, so it is going to be present over here. Then if I talk about the plasma cells, so plasma cells, obviously plasma cells produce the antibodies, so the plasma cells are obviously going to be there in the present in the primary response to the virus. And if the plasma cells are there, so obviously the beta lymphocytes, B lymphocytes are also going to be there. Why? Because B lymphocytes basically divide itself to form the plasma cells. So the B lymphocytes would also be there. Also if I talk about the T helper cells, so obviously T helper cells are the king of the immune system, and so the T helper cells are going to release the cytokines, and this is their main function. Basically the T helper cells, the main function of the T helper cell is to release the cytokines to activate the other immune cells. So obviously it is also going to be present during the primary immune response. Yes, if they would have asked that what about the memory cells, are the memory cells going to be present during the primary immune response? So no, the memory cells are not going to be present in the primary immune response. However, in the secondary immune response, the memory cells are going to be present. But in the primary immune response, the memory cells are not going to be present, right? So this is how you are supposed to answer this part. I hope that this is clear. Now let's quickly move on to the part C of this question. Part C of this question says that the cell cycle can be divided into different parts. Complete the sentence A, B and C to provide more information about the mitotic cell cycle. Part A over here says that the part of the cell cycle that occurs immediately after the mitosis is. Alright, so for this let's refresh our knowledge about the cell cycle over here. Now this is the cell cycle over here. If I talk about the cell cycle, so this G1 phase, S phase and the G2 phase, this is basically the interphase of the cell cycle. And then from the prophase, metaphase, anaphase, telophase, this is the mitosis phase of the cell cycle, and then there comes the cytokinesis phase over here. Now they are saying that the part of the cell cycle that occurs immediately after the mitosis, so that is the cytokinesis. Cytokinesis is a phase that occurs right after the mitosis. And what is cytokinesis? Cytokinesis में हमारे पास क्या हो रहा होता है? The division of the cytoplasm to produce the two identical daughter cells is basically happening in the cytokinesis, where the cytoplasm of the cell is basically dividing to form the two identical daughter cells. So the answer over here is cytokinesis. And then if I talk about part B over here says that the part of the cell cycle in which S phase occurs. Alright, so I told you guys that there is a G1 phase, there is an S phase, and there is a G2 phase. These phases all together are known as the interphase. So the S phase basically occurs in the interphase. Interphase हमारे पास क्या होता है? Interphase is basically the period before the mitosis actually takes place, where the cell grows and it prepares itself for the cell division. Now in part C, they are saying that the main event that occurs during the S phase of the cell cycle. Now in the S phase, what is actually happening in the S phase? There is the DNA replication, that is the main event occurring in the S phase. Each chromosomes will form the two sister chromatids, which are going to ensure that the identical genetic material is being passed to the two daughter cells and with the same number of the chromosomes as the host cells. Alright, so now this is basically how we will answer question number 3. I hope that this question is clear to all of you.